found a way of stopping the growth of GBM, glioblastoma cells, tumor cells, which were derived from patients with a specific drug. And then when we tested it in animals, in an animal model, we found that the tumors didn't grow as well. When the drug was used in combination with current drug that's already used in GBM, for treating GBM, it's temozolomide. The two drugs together significantly extended the lifetime of the animals. The next phase of this study, which originated in the laboratory, is to use and test these drugs in patients in a phase 1-2 clinical trial for GBM in Canada. We've been working on this specific project on targeting these cells for a number of years. With this drug, it's been about 2 to 3 years that we have done various tests on cells in the laboratory. The breakthrough really came when we saw significance in animal studies. Basically, it's a model for the tumor in humans, as close as you can get to a human without actually using the human yet. So that was the breakthrough when we saw the extension of life in combination between the two drugs. So the current drug that's used in clinic, temozolomide, TMZ, actually introduces damage in the cells. If the cells have a protein called MGM2 protein, they are able to repair that damage, and the drug doesn't work so well because the cells don't die. The damage is repaired and the cells, cancer cells or otherwise, can go on and keep growing. If the cells don't have this protein through a mutation in the DNA, then the temozolomide works. It introduces damage, the cells can't repair that damage, and the cells end up dying. This, in patients who don't have the protein, temozolomide will work for a while, but eventually the cells adapt, and because they're tumor cells, so they find ways around this, and the temozolomide doesn't work anymore. With the EGFR version 3, what you said, it's some patients have this specific protein. It's an alterated version of the EGFR protein, and it is thought that some of the patients who have, or some of the cells that have this protein, are a little bit more resistant to current treatments. There have been drugs that have been used to target these proteins directly, or these cells directly, but eventually they end up not working so well. Mostly because perhaps the cells have a way of adapting to these various treatments. The AZD8055 targeted a specific process in the cells which is responsible for growth and proliferation, so the cells for the cells to divide, and then the TMZ induces the damage in the cells. So in combination, these two together resulted in the cells dying, and the cells not being able to repair and to continue to grow. So it seemed like it was particularly effective even in cells that normally had the MGMT protein, which, if just with TMZ alone, would not work. The AZD 2014 is a direct analog or derivative of the 8055 that was produced by AstraZeneca to improve the side effects of the drug, so to make it more useful for human use. When a drug is created, sometimes there's unwanted or side effects that they don't always anticipate. So with the AZD 8055, they found that while the drug is still effective at killing the cells, that perhaps there are other side effects that are not so good for human use. So now there's a new version of the drug, the 2014, which is addressing some of those concerns. And that's the drug that's actually going to go in the clinical trial for GBM.